My name is Dror Minz. I'm a researcher at the Volcani Institute, which is the R&D unit of the Ministry of Agriculture in Israel. And I'm at the specific Institute of Soil, Water and Environmental Sciences. I'm a microbial ecologist, and I want to use this opportunity to convince you that microbial ecology is not a bad word. Ecology, usually when I say I'm dealing with ecology, a lot of the people around me would say, you are hugging trees and you are doing things that are non-relevant to the common people, and I want to try to convince you that it's not the situation. So what I'm going to talk about today, I'll introduce you to root microbial community composition, what we call microbiome. Most of you heard of human microbiome or gut microbiome. Microbiome is the microbial communities in any specific niche. And in my lab, we are doing mainly plant root microbiome studies and also some soil microbiome studies. So the second thing I want to show you is not only the composition of the microbial communities, but also the function of the microbiome in the plant root, which means the communication and support of the bacteria to the plant and vice versa, the plant on the bacteria. Just to let you know, bacteria, the plant can secrete from the root up to 40% of what the plant fixes, the carbon that the plant fixes, and this is in order to support its preferred community. And the third thing that I want to show you today is how we can learn from the root microbiome about the conditions in the niche. Once again, the niche will be the plant root microbiome and the plant root as a niche. I want to show you how we can learn how the microbiome can teach us about the conditions in the soil surrounding the root. And then I will show you an example of how the microbiome of the root can function not to the best conditions or malfunction of the microbiome in the roots. And after that, I will show you how we can use this understanding, the know-how and the understanding of the microbiome to solve an environmental problem. So quite a lot of years ago, a student by the name of Maya in my lab studied the community composition on the roots of 4 different plants. We chose 4 plants that are important for consumption by man: cucumber, tomato, maize, and wheat. In this figure, what you can see in the y-axis, the relative abundance of different communities or different taxa, and the names on the right are not important at all to our meeting today. What I want to show you is just that the 100%, we never get to 100%. What we do see is that there is hundreds of populations that are in such a low relative abundance that do not allow them to get into the figure. So in the cucumber, for example, only less than 35% of the abundance is by bacteria that are above 2% in our case. So there's enormous diversity that is hiding here. And on the other hand, in the cereals, we see a dominance of one organism, labeled blue here, and this organism is the most dominant organism, and the rest of the diversity hiding here in the red arrows is very small. So we have two contradicting ecological situations. One represented by the cucumber with an enormous diversity, and the other represented by the cereals with less diversity and much higher dominance of this organism. I'm not going to go into names of the bacteria. You will not convince me to give you even one name of bacteria in this talk because it's not relevant to most people. Next, we wanted to see we took the two extremes: wheat as representing the dominance, and cucumber as representing the high diversity. And wanted to ask more questions about the interaction with the plant roots. And more specifically to this talk, we compare the metagenomes, so all the genomes of all the organisms in the plant root, and we are talking about many thousands of different species in plant root, so many thousands of different organisms, and in each of them many thousands of genes, so it's an enormous amount of data. And these are the genes and also the metatranscriptome, which means to look at all the genes expressed by the community on the root microbiome. Here we have the cucumber communities and the wheat communities, and we ask the question, are the genes expressed differently by the root microbiomes on different plant types? So in the study I showed you before, this one, the plants were growing in the same soil, in the same potting system, in the same growth chamber, by the same student, irrigated by the same water, and I was also the same, but much younger and darker hair. So all the differences that we're going to see now are due to the interactions between the plant root and the microbiome in the soil, and enrichment of specific members of the community in the soil, making them community of the root. Okay. So I'm trying to make a very long story short, and this figure represents a theoretical cell, and this is the genes expressed by this theoretical cell that represents all the community in the cucumber and in the wheat system. We dyed green the genes that are expressed in high level in the cucumber system, and in yellow genes that are expressed in very high level in the wheat system. And I'm not going to go, and these are the most statistical differences. I'm not going to go into details, and I will even hear that this very, very simplistic way of looking at the expression of the genes. Even here, it's too complicated to explain in a short meeting like that. So I will show you one thing. One of the interesting things is in the lower part of the figure, you see molecular symbols representing organic matter produced genes that are degrading organic matter, and you can see, for example, in the lower left, mannan and pectin and starch and silane. So these are genes expressed in high level in the cucumber system and not in the wheat system, suggesting that the bacteria in the community, while they are on the root of the cucumber, probably exposed to those carbon sources, therefore express genes to degrade it and consume it for their growth. In yellow, you can see, for example, arginine in the lower right side, which is probably very common in the wheat system. Another interesting thing is that when we look at the cellulose, the two communities express differently enzymes that have to do with degradation of cellulose. Exoglucanases in the cucumber system and endoglucanases in the wheat system, suggesting that the branching of the cellulose molecules is different in the two systems. And this is, I'm not going to go into that because this is not my main interest in the talk. I just want to take you look at ask you to look at the red square on the right, where you can see genes that are expressed highly in the community on the wheat system, on the wheat root system, and mostly the dissimilatory nitrate reduction. These are genes expressed that have to do with anaerobic respiration in the presence of nitrate. So nitrate, NO3, is available in the irrigation system where the plants were grown on, and the fact that the community, not only one organism, many different organisms in the community in the wheat system express to high level these genes suggest strongly that the community is exposed to lack of oxygen. A lack of oxygen can happen because of aerobic respiration that consumes root exudates and available oxygen to a situation where the level of the oxygen drops down and the community sense lack of oxygen, therefore expressing the what we call denitrification pathway genes. Another thing I want to show you is that so when we look at closely at this pathway, there are many different enzymes involved here. So nitrate is reduced to nitrite, and then we have that reduced further up to nitrous oxide, N2O. You know N2O from the dentist. This is the laughing gas, or it's also called, I forgot the other common name for it. This is the gas that you breathe at the dentist to make you less nervous before the dentist starts drilling and before it gives you the check. So nitrous oxide is the product, and what is now missing is the last gene that is probably not expressed at high level in the system. The gene is called NOS-Z, and the enzymes should reduce the nitrous oxide, the N2O, to dinitrogen N2, which is an inert gas that is released to the atmosphere if everything works fine. So we know now that there is a problem. The community does not express well the NOS-Z, and we ask a question. We ask ourselves, can we use this data to make any difference? So this is like ecological study with ecological meaning, but can we use it for something? So I ask you to look a little bit at the denitrification. I said it's an anaerobic respiration process, and it's the most important producer of nitrous oxide. And this is the whole process. And nitrous oxide is actually the villain in the story because it's a very potent greenhouse gas. It's up to almost 300 times worse than CO2 as a greenhouse gas, and it's also damaging the ozone layer. So it's really a bad gas, and we don't want to have it emitted from the soil to the atmosphere. And actually, the study by the US EPA showed that, so we know that up to 40% of the emission is anthropogenic, meaning man-made. So 60% or so is natural, but 40% is caused by a human. And out of this, 3/4 in the dark blue part of the pie, 3/4 is due to agricultural soil management. So we hypothesized then that because of the fact that the microbial community in the root system in wheat and probably also in other cereals, at least, is not able to reduce nitrous oxide into dinitrogen because of the fact that the nitrous oxide, the NOS-Z gene is not expressed in high enough level by the community, may be a cause for the emission of nitrous oxide from soil agriculture. So the question we asked then is, can we minimize the emission somehow? And there are two options of theoretical options of minimizing the emission. One is to prevent the production of nitrous oxide. I don't know, maybe killing all the bacteria that produce it or something like that. This is impossible, because I told you that in the root community we have many thousands of different of different taxa, different bacteria, and then even if we kill 99.99% of the community, there will always be some member that will survive and take over the system and will continue to release nitrous oxide. So the second option was to consume the nitrous oxide before it's leaving the soil into the atmosphere. And this is a story that Ala Osishkin in my lab, that did a PhD and then a postdoc, took upon herself to find a way, a microbial way to consume the nitrous oxide before its emission. So how did she do that? She went to the wheat roots and then homogenized the roots, plated the solution on agar plates, put them in a desiccator, flushed all the oxygen, introduced nitrous oxide as an atmosphere into the desiccator, allowing to bacteria that can reduce the nitrous oxide to grow on the, this is nitrous oxide introduced into the jar. So the desiccator is completely anaerobic, and the only electron acceptor for the bacteria to grow on is nitrous oxide. And then we got bacterial colonies and analyzed their activity as nitrous oxide degraders or reducers, sorry. It looks very simple in this figure, and in matter of fact, it's even more simple than you can think. It's very straightforward. Every microbiologist can do it in any lab, basically. So Ala isolated a few. I'll show you 3 organisms that she studied. Isolate 14, 243, and 128. And what you can see in the figures here, that each of them, when closed in a bottle where the atmosphere is lacking electron acceptors but nitrous oxide, the bacteria consume the nitrous oxide, and in the blue line you can see a decrease in the concentration of the nitrous oxide in the atmosphere of the jars, and this allows the bacteria to grow to higher biomass. So in blue we have a decrease in the nitrous oxide, and in return we see growth of the bacteria in brown. So the bacteria can definitely reduce nitrous oxide, and then of course we did the genome analysis, and what you can see is that all 3 of them have in the right side the NOS-Z gene as expected, because this gene NOS-Z is the only gene known on Earth that can reduce nitrous oxide to dinitrogen. So they all have the NOS-Z, and then we went along and did probiotics on the plants in order to minimize nitrous oxide emission. We grew the wheat, amended the bacteria. This is exactly how they look under the microscope. We amended the bacteria into the soil in which the wheat grew, put the pots with the plants and the amended bacteria. Just the important thing to say, that the soil is natural soil. It's not a sterile soil. It has all those hundreds of thousands of different soil species, and we added our bacteria in excess over the natural bacteria in the soil. So when we closed it in the jar, the bacteria, the microbiome, the natural microbiome produce nitrous oxide by the process of denitrification, and we measure the concentration in the atmosphere when amended with bacteria, our specific bacteria. So this is how it looks in real life. These are glass jars. We have a septa, and a syringe was used to take out atmosphere, and the results are shown here. We did it in two different conditions, one which is anoxic and one that is ambient. So in the left part, we removed the oxygen. In the right part, we did not remove the oxygen. And then we see this is the anaerobic condition where we remove the oxygen by flushing it with dinitrogen, which is irrelevant to the story. And in the y-axis, you see the emission of nitrous oxide into the atmosphere in ppm, parts per million. And the level 100 ppm is very high because this is an anaerobic condition. In an anaerobic condition, we favor denitrification. As I told you before, this is anaerobic respiration. So when you look at the emission from the other pots, and the number above the bars is the number of different pots, the replicate number, and you can see the control had 81 different pots with its own plants. And in the other three, you can see smaller number, lower number, but still, you can see that bacteria number 14 decreased the emission a little bit. 243 lowered the emission better. So it's now statistics, as you can see with the asterisks. 128 will always be bad. It's a bad organism. It's not efficient in reducing the nitrous oxide. Even probably here, we can probably see production of nitrous oxide. Okay, so this is an anaerobic condition. In aerobic condition, as denitrification is not so popular by bacteria that have oxygen, the control have about a little more than 30 ppm. Bacteria number 243 was not efficient. 128 was bad again, but bacteria number 14, isolate 14, lowered the emission by approximately 50%. So in pots, the bacteria did what was expected for them. They reduced the nitrous oxide in the soil, in the root system, minimizing the emission by up to 50% in some cases. So next thing happened, people with money came along and invested, and we have now a startup company that is supposed to produce a product which in our vision will have the product will have one or two bacteria capable of reducing nitrous oxide emission from soil by at least 40%, with a long shelf life in the can for the farmers to apply whenever they want to, and a very high concentration of bacteria. And we aimed at wheat and corn, that we think is the most important products, the crops for this product. So we did now as a company, we do a lot of different studies. For example, we wanted to see how the bacteria operate compared to dead bacteria. And the question we ask here is the amount of bacteria applied to the pots important. So we applied a specific concentration, 25 milliliter, 12 milliliters, 6 milliliters, or 3 milliliters, and there is a slight improvement at the 25 ml, but this is compared to the control that have dead bacteria as an organic matter that is added to the soil in the form of bacteria that may die and degrade and provide nutrients. So if you look at each of the concentration in the left bar, you see that the levels are all very similar. So it's not important how much bacteria we actually amend. And I think this is because these are root organisms. We isolated them from roots, they colonize the root, they grow and multiply, and therefore it's not important how much we apply, they will eventually get to the high enough level. So let's continue. Another thing we wanted to see is it enough to apply once, or should we apply twice during the growth season of the cereal? And you can see in the left figure, once again, dead versus life. And in all cases, the life bacteria gave very similar results, always very significant compared to the dead bacteria. All of them are approximately half of they removed half of the emitted gas. So let's continue. This was done in a very small pots until now, and then we wanted to see how they behave in much bigger pots. We didn't have a large enough glass jar, so we have to improvise. So we grew the plants after amended with bacteria for 15 days, and for in the upper green figure in the upper left side, and for 30 days. And then we looked at the production of gas, and it's all doing the same. So basically, when we went to a much higher soil content, many more plants in the pot, we can still see the efficiency. And then we'll check the amount of time post-amended of the bacteria, and we can see that it doesn't matter 15 days or 30 days after the amendment, we still got a very efficient removal of the nitrous oxide, and this is done now with 2 different soils. So we wanted to see more soils. And then we said, okay, should we apply the bacteria in solution to the seeds, or to the root system, or to the soil, or can we coat the seeds and let the farmer sow the seeds with the bacteria already? And once the seed will germinate, the roots will emerge, and the bacteria will colonize and grow. And here we can see, once again, bacteria 14, when we applied on the seed dead bacteria in the left side, we had 75 nanograms of nitrous oxide per gram soil, and almost half of it is not emitted when we apply on the seeds live bacteria. And then I think my time is running. So the next thing we did check is whether corn is also a platform. So we checked the bacteria, I remind you, isolated from wheat roots. We checked it on maize, on corn system, and the results are the same. In the left bars, we have the dead bacteria. In the right bars, the live bacteria. And we again get 50 or so % decrease in the emission. So next thing we did is try to find better bacteria. So is more the better? More bacteria is better than less bacteria? In yellow, in orange, or I don't know, maybe you see it brownish, is the bacteria we did all the experiments so far, bacteria number 14, but and it's removing, so it's removing just like all the other studies we did, but we have in the left of it bacteria that removed even more nitrous oxide from the gas emitted from the soil, and some of them are producing spores, and this is something we aimed at, because if the spores are much more resistant to the environment, the product will be much better. So we have now bacteria that are much better than the ones we started with. And what are we doing now? Right now, as we speak, we have a field within Vulcani where we have huge containers, huge for microbiology, okay? Containers filled up with 4 different soils. It's outdoor, it's not in the greenhouse, it's not in the growth chamber, it's outdoor. The temperature is very high. and we applied the two different bacteria, one of the spore formers and one number 14 that I showed you most of the results before. And now we don't use the jars anymore, of course. We apply a small collecting device on top of the containers. This is a container, the zoom on one of the containers with the corn plants. This was taken today. Okay, so it's really very, very fresh. These are the corn plants. They are a week and a half old. And this is the machine that measures the emission of the nitrous oxide using a specific laser. The machine is called FTIR. And we are now able to measure in the field the different, we have 128 different containers here with all the combinations and controls. So to finish, I think my conclusions are, first of all, knowledge is power. If we didn't do the experiments, the ecological experiment, we didn't have the question to ask, and we didn't know that there is a malfunction of the community, and ecology is knowledge, therefore ecology is power. And I could show you that using the ecological tools, we can actually make a difference. And if we succeed in the container study, in the winter, we now plan to do a real field experiment with wheat, in which we check all those conditions in a real agricultural farmland field. And if this succeeded, I'm not expecting 50 or 60% reduction in the emission, even 5%. If we succeed to decrease it by 5%, your grandchildren will appreciate it, I think, because, as I said, it's a very problematic gas. I want to thank you very much for listening. I want to thank Rush University and especially Stefan Green for inviting me to give this talk. And if you want to find me up on web and send me questions, I'll be happy to answer. And I hope you have an excellent conference.